was thought before because of Sarian and Nathan. We joined our research team to conduct this section here to talk about fish ponds, right? And yeah, Sarian works at Radboud University. You probably saw her presentation during the Shallow Lakes meeting. And Nathan works in the Federal University of Juiz de Fora in Brazil. And I work at the University of the State of Mato Grosso in the Pantanal, right? And when you see this this biome, some people, they don't understand what's the Pantanal. I used to say that's just below the Amazon, but it's more beautiful than Amazon, right? And here I live and here I conduct my work. Thank you everybody to be here in our session. And I just would like to share the screen with some thoughts just to introduce this topic, right? And then we pass to the discussion. If Nathan or Sarian or someone wants to say something before, please feel free while I share the screen. I don't know if you're seeing the screen. Please let me know.
Yes.
Yes.
Yes. Thank you very much. We are in the session 16, already 4 days of the beautiful conference that was supposed to be in Brazil, actually is in Brazil, right? And thank you very much, Mariana and Priscilla, to organize everything here. And yeah, of course, would be much better if we were in Natal, at least smelling the sea. Smell is really good and the beaches are very nice, but okay, we need to go, right, for these online sessions, and it's been very nice. In this session 16, we talked about to talk about fish ponds, right? And first of all, why fish ponds? And this is the abstract of the session. Why fish pond, right? Why? And then we thought, no, fish pond is the literal translation of shallow lakes, right? Because it's shallow and it's not so deep. You cannot maintain a fish so much deeper in the water. And then we go for these fish ponds that are a clear shallow lake translation, for example, right? And when you start to search about fish ponds in the literature, in the websites, for example, you find so many papers nowadays. And these fish ponds, the people that are researching about, conducting research about fish ponds, they are talking about microplastics in fish ponds. They are talking about limnological variables like oxygen or pH or conductivity and many others. Also about nutrients, nitrogen, phosphorus, for example. And we also find a lot about sustainability and what's the main challenges about fish ponds, and what about the people that are working with fish ponds? Is it sustainable? It's environmental sustainable. It's social sustainable. But also we find quite some, not so many papers about greenhouse gases, right? But we start to find this in this few years ago. We start to talk about these greenhouse gas emissions from fish ponds, and some of the people here from this section, we published a paper last year about the assessments of greenhouse gas from global fish ponds, and was a research conducted from Sarian and Nathan, Raquel, and some people here. Renske was also together, right? And then what I did yesterday, just to give an introduction of the topic here, I just put in the Science Direct how many something like fish pond. When I put the fish pond there, the fish pond together, the word together, fish pond. When I put there, and you can see here, let me check if I can take another pointer. Yeah, when you see here in blue, I think it's blue, yeah. The blue one, you see around 150 publications the last year with the word fish pond. For me, it's already quite a lot. And then when you see the word split, it's fish pond, but in between, yeah, how to say aspas? Forgot. Yeah, and then you see quotations, right? And then you have around 300 publications. And then in the last year, you have around 450 publications working about fish pond. If you put aquaculture in this word in the Science Direct website, you will find around 9,000 publications. Of course, when you say aquaculture, you also talk about marine and other kind of productions, right? But you see, fish pond is already a hot topic. That's why we try to make this session, conducting this section about this topic. And what we found is that in FAO in 2016 showed this beautiful report showing that the food supply from aquaculture is already much more, or 2 times more, than the first fish supply from capture fisheries, right? And then what we see here is that the aquaculture, or linked to the fish ponds, is really a topic that we need to think about, that we need to discuss about. And this aquaculture ponds, or the aquaculture production, it's increasing a lot from the 50s to nowadays. It increased like 80 times from the 50s, while the population only increased like 3 times. Then if you divide, we have something like 20 kilos per person per year of fish produced in aquaculture ponds, right? And this is really increasing. That's what we see here. And of course, when we talk about aquaculture or fish pond, we go to China, or we go to Asia, right? And Asia is the main region producing fish or working with aquaculture in the world. And China is the most responsible for this amount of production, followed by Egypt, India, Indonesia, and Vietnam. And the fish that are mostly produced are carp. When we put all the species of carp together, we have around 37% of the global fish production, And then this means that carp is the main fish being produced in aquaculture in the world, right? But only 92 countries reported that they are producing carp. When you go for tilapia, that's the second group of these species that are produced in the world, only 7% of tilapia, tilapia represents only 7% of the global fish production in the world, but 126 countries reported that they are producing tilapia. And then we have those two groups of fish, carp and tilapia, that are mostly being produced in the fish ponds in the aquaculture world. But with this such amount of production that's increasing a lot, we also have some issues, or as how I like to say, we have some challenges, right? And one of the most challenge that we found for now is the feed input, right? When we put some feed for the fish or for other cultures, you have an input of nitrogen, of phosphorus, and carbon. And the feed input is also one of the major challenge that the producers report, because it costs money. It's quite expensive to feed the fish, and also is increasing a lot the value of this feeding. And then when you put lots of nitrogen, phosphorus, and carbon in the environment, of course, some part of this is ingested by the fish. Around 35% is ingested for the fish, and part of it is dissolved in the water, and part of it is, or greatest part of this, is accumulated in the top of the sediment, right? And this accumulation can also turn the water quality into the bad water quality, right? Causing eutrophication, for example, reducing the fish yield, and sometimes also causing fish kills. And this is very common. We see this quite often, at least here nearby, when we have these. Of course, when we have lots of technicians taking care of these fish ponds, it's much more difficult this happen. But when you have some people that they don't have so much money to have new techniques or new equipments to take care of the water, and then you have these fish kills, and it's very common to see here in my region. But also, when this such amount of carbon or feed is accumulated in the sediment, it can be turned to greenhouse gas, for example, to methane, to CO2, or to N2O. And these gases produced in the sediment or produced in the water column, they go to the atmosphere by diffusion, or by ebullitive transport, right? And what we need to highlight here is that when we produce methane and N2O, for example, they are very potent greenhouse gases. That methane, for example, is 34 times more potent for the global warming than the CO2. And N2O is even worse, it's 298 times more potent. And that's something that we need to take into account when we go to conduct some research about pisciculture or aquaculture or fish ponds. But then what we need to think a little bit more, it's with the species that we have in the inside of these fish ponds, right? We have some nectonic ones, we have the bentivorous ones, we have different species of fish, some that need more feed, some that need less feed. We have crabs, for example, that's Junji work. We have macroinvertebrates in this environment. And this is what we brought to this section now, to discuss about which kind of systems we are talking about, which kind of fish ponds we are aiming to have, which kind of sustainability we want to have with fish ponds, with more greenhouse gas, with less greenhouse gases, which kind of what's better for the environment. When we put aeration, for example, we remove the aeration. We have the fish ponds drained or not drained. And this is something that we want to discuss. And yeah, that was a very short introduction about our topic. And then we open the floor for what are the researchers doing around the world. We have here in this session 3 presenters. We have the presentation for Marcelo, for example, and it's talking about fish farming in tropical reservoir and sediment bioassessment and greenhouse gas emissions, right? They conduct a research in Ilha Solteira. Ilha Solteira is next to Mato Grosso do Sul, right, Marcelo? I guess so. Yeah, it's between São Paulo and Mato Grosso do Sul in Brazil. And they conduct their research in the net cages, right? And they went to the reservoir and they sampled sediment and they saw the greenhouse gases and also the macro invertebrates community. Marcelo, do you want to talk a little bit about your topic? Would be nice if you want.
Hello everyone. This study, it's we have 2 parts: the greenhouse gases and the biological part. So I will present the greenhouse gases, and then Mariana will present the part of the biological part. So Mariana, can you please show? I don't know what screen are you seeing? It's not ours. Okay.
What do you want from me, Marcelo? I didn't understand. Sorry.
I think that you can start the presentation. So.
Okay.
Sorry, I think for starting the presentation, we need to make them as hosts, right? Mariana or Priscilla, I don't know if you're still here.
I think you're going to have to stop sharing your screen, and they should be able to share it too.
Yes, I have to change the presentation.
Okay, I stop sharing.
Now see, Mariana, see if you have access to something called share screen at the bottom.
Okay. Yes, I have. Just a moment, I'm going to open the presentation.
Ernandes, maybe it's good to explain a little bit the dynamics of how we would like
Okay.
to do it, so everybody can pitch his presentation in a couple of minutes. So,
Okay.
and then we can ask questions afterwards.
Yes. Do you see the presentation?
Yes.
It's perfect.
Okay, so this work, as Marcelo said, it was conducted in two parts. We had the biomonitoring of sediments, including the benthic macroinvertebrate community as bioindicators, and the effects of the greenhouse gases. So the support capacity of Ilha Solteira Reservoir is about 121,000 tons of fishes produced, and it belongs to a project called BRS Aqua, which is a big project where many units of Embrapa are involved. And its goal is to develop research infrastructure and generate and transfer technologies in aquaculture, considering the sustainability of the activity. So the biomonitoring, as you know, assess the impacts on biodiversity and changes in water quality, directing environmental management in natural and artificial aquatic ecosystems. And the benthic macroinvertebrates had a great biodiversity, including tolerant and sensitive species, and they act directly on the nutrient cycling and pollutant release at the sediment-water interface. That's why we tried to link the bioindicators and the greenhouse gases. So here we have some examples of benthic macroinvertebrates found in Ilha Solteira Reservoir. And the main taxons are bivalves, like Corbicula fluminea, for example, and the gastropods Ilacostoma tenuilabis in the right side here. And we also have some leeches, like Elobidella, and some aquatic insects, larvae, like Abablesmia, for example, is a genus of Chironomidae family. For the greenhouse gases, I don't know if Marcelo wants to talk, but we measured methane, carbon dioxide, and nitrous oxide. And these gases they are produced due to organic matter from uneaten feed and fish feces. And the main sources are these ones. And the gas production is a deposition of organic carbon that can be the source of the production of greenhouse gases, according to literature. So we had this objective that was to assess the aquaculture activity in the Solteira through biomonitoring, using the benthic macrovertebrate communities and the emission of greenhouse gases in two distinct areas, with which have expressive fish productions in net cages. So as Ernandes has shown, this is the reservoir, the Ilha Solteira Reservoir, and it's located between Mato Grosso do Sul and São Paulo states. And we worked in two river branches of where we are concentrated the fish production, the Formoso River and the Cancan. And sorry, And here at the right side, we have a photo of the net cages where fishes are produced. So we had 3 field campaigns in 2019, also, in June, in February, June, and September. And we used the GRAD, the Ekman dredge, and divided the area, the production area, in 3 regions, in 3 sites. Upstream, the production area, inside the net cages area, and downstream the net cage. We had some the measure of organic matter content, percentage of organic matter, the concentration of metals in sediments, and also for greenhouse gases, the carbon concentration in the sediments, and limnology parameters measured by multi-parameter probe. And also in the laboratory, the sediment was analyzed and the benthic organisms were identified by morphological and molecular taxonomy using the COI genes to the molecular identification. So here is a photo of the samplings, the sediments.
Mariana, I'm sorry to interrupt you because we want to discuss a little bit more about your research. And if you go onward to your main findings, for example, would be nice, because then we can go for the discussion afterwards. And then the other presenter, sorry, I did not say this in the beginning. I just sent by email that the participants would have between 2 and 5 minutes to discuss or to give a short presentation. That's no problem, right? We have sometimes you, but if you move onward to your main result, main findings, would be very nice. Thank you very much.
Okay, so we have here the main results of benthic macrovertebrates, where we can see in the left side the graphics of taxonomic richness and channel diversity index. We had lower values in the net cages, that's in the middle of the figure. And in the opposite, in the right side, we have the density of organisms and the PLU evenness index, where we also have had lower values of evenness PLU index. And so the figure shows us that where we have the net cages area, the metrics of biologic bioindicators are lower, indicating there is probably an impact, a local impact in these areas when compared to upstream and downstream of net cages areas. And the same result we found in the Formosa River, where the richness are lower in the net cage areas and the diversity. And so in the right side, there is a great diversity of organisms in the net cage areas, much greater than the upstream and downstream areas. Do you want to talk about the gases, Marcelo?
Yes, thank you, Mariana. To greenhouse gases, what you saw is that for diffusive methane, we had a significant difference between the controls. The controls was the area before the tanks, the net cages, and after the net cages, following the water flow. So we saw this increase of methane, diffusive methane, in the tanks, in the area of the tanks. And, we didn't saw the same behavior to CO2 and nitrous oxide. So this is a first impression, a first what I say, that we have influence of the net cages.
I think his internet is.
Yeah, I think his internet was lost.
Okay.
Can you still continue, Mariana, or?
Yes. As he was showing, the there were a significant increase of diffusive methane in the net cages, where you can see in graphic A. And for CO2 and nitrous oxide, there was no significant difference in graphics B and C, respectively. For ablative gases, we had a significant increase of methane in net cage sites, as we have in the graphic A, but there was no significant ablative between sites for dioxide, carbon dioxide, and nitrous oxide for diffusive fluxes, as we can see in the box plot figures. And for diffusive methane, we had an increase of diffusive and ablative methane fluxes. The carbon concentration in the sediments was greater than 4%. And carbon dioxide and nitrous oxide diffusive and ablative fluxes, there was no influence of carbon in the sediment, according to the results. So for conclusions, we saw that the ecological attributes seem to have been affected by fish farming, as we saw that the species richness and minor diversity in the net cage areas, which suggests changes in the balance of benthic communities as bioindicators. However, it seems that this change is localized and this does not compromise the water quality for fish production, at least at the study area, because we saw water quality parameters that attained the limits for CONAMA resolution. So the fishes were well. There was not any damage to the fish production. And the analysis of the results of greenhouse effects suggests that the production of tilapia in the Solteira reservoir influences the methane fluxes. But there was no observed influence for carbon dioxide and nitrous oxide fluxes. Thank you very much for your attention.
Nice, thank you for your beautiful presentation. Nice.
Thank you.
And Nathan has a question already.
Yeah, thanks, Ernandes. Thanks, Mariana and Marcelo. Is Marcelo back? Maybe he's having problem on his connection. But it's very nice to see your presentation, Mariana. And what calls my attention is that maybe your conclusion for biological community can be applied for the greenhouse gas emission as well, because it seems to me that the effect exists for methane, but it's a local effect. Based on your data, I don't see like influence of the fish ponds or the cage upstream or downstream of the area of production. So I want to ask you, like, if this is right, if this is what you found, and also how you explain that, like, the local effect on greenhouse gas emission.
Okay. Oh, you are right. We observed no impact or no community alteration in the upstream and downstream areas. And also we had a sampling in rivers, which represented different areas. There was no, it was far from the production areas. And we had greater values of channel diversity in these rivers. So we expect that is a local impact, and we relate this on some species like Oligochaeta, for example, that benefits from organic matter, the increase of organic matter, which are located in downstream, close to the net cage areas. So we are now trying to make statistical relationships between some specific taxons that we know they tolerate low organic dissolvent concentrations and anoxic conditions, and relate the emissions of methane with these specific taxonomic units. Did you understand?
Yeah, I do. and I think it's quite hot because some people agree that the effect is in general large, and then you show us. So if you can show that it's a local effect also for greenhouse gas emission. I remember that I saw some pictures, extra pictures related to this work, and it was clear that the effect was lower than local, and it's completely different what the literature says about it. So thanks, yeah.
Yeah, just
to complement, sorry. Oh, Marcelo's come back.
Yeah, I'm back. Sorry. We only see this effect of the net gates on methane emissions. We didn't see to CO2 and nitrous oxide. So the relationship, the most important variable to explain this variation of methane was the carbon in the sediment. To carbon dioxide and nitrous oxide, we saw a different relationship with temperature, another compounds of nitrogen in the water. So the methane was the only gas that showed this relation to net gates and the carbon in the sediment. And this emission decreased far away from the tanks. So because of this, we conclude that this influence of the net gates was local. The ambient has this, what? Sorry?
No, I got it. Just one more question. Do you also have, can you tell me about the oxygen in the reservoir and also the eutrophic state of the reservoir? I'm just thinking about the implications of this work. Is it like a reservoir with low oxygen in the water column, far from the fish cage, or how is it?
You want to answer, Marcelo?
I can remember specifically the oxygen. So do you remember, Mariana? I remember something like 5 milligrams per liter in the area.
It's not so bad. It's not so bad, the solid oxygen concentrations. In the net cage areas, I observed sometimes lower than 5 milligrams per liter. So okay. It's lower than the other areas, but it's not so low that compromises the development of fishes. No. So and also we have to pay attention in the management practices. For example, it was not the density of fishes were not so high to compromise the water quality. So that's why I think we found local impact, because and this impact is not so great for these reasons. Okay.
Sarian has a question.
Yeah, thank you very much first for this very nice and interesting presentation. I also had some questions, and one was more a remark, but one question was, I mean, these are net cages that are situated in a much larger system. Would you expect more than a local effect, especially if they are just not there for many decades already? I think it may be expected, right, that it's local.
Yes, in areas where we have greater production of tilapia, we hope that this impact would be greater. And sometimes the eutrophication, where the eutrophication is bigger, the producers change this area from a better condition of water quality. We saw this happen, okay, in some cases. Yeah. But in general, Ilha Solteira had great conditions to develop the aquaculture.
And do you have some idea about how much carbon is being built up in the, I mean, I guess the pollution is building up in the...
I think Marcelo is better than me to answer this.
Yeah, can you repeat, please, the question?
So you see higher organic carbon below the cages. So
Yeah.
can you say something about how much is building up there?
I can remember, but I can remember some numbers. I like 6 or 8% of carbon in the net cage, and this value decreased 1 kilometer ahead of the tanks. So we need to do another tests, another studies in the area to see this contribution of the carbon, how much is this accumulation of the carbon. But this is a first study, so I'm not sure I'm not have these conclusions to talk to you now.
Thank you. And then one is not really a question, but a remark that I think is very nice, and it relates to some experiments that we have been doing. If the fish cannot enter the sediment, like in your case, of course, there will be wild fish as well, I assume. But in that case, we also had a lot of oligochaeta in the sediment, and they oxygenate the sediment much more than when there's fish present and predating on the oligochaeta. So we also found that when these benthic organisms were present, we would have much less methane being produced than you would expect based on the organic matter content. So I think it's very nice. It matches very nice with your findings as well.
Mm-hmm.
Thanks.
Thank you.
Thank you.
Thank you, sir.
I think Ikaru has a last question, then we move onward to Renske's presentation.
Okay, hello everybody. Hello Mariana and Marcelo, thanks for the nice talk and congratulations for the presentation. I'm curious about the methods of the methane emissions. You said in your presentation that you use a gas chromatograph to analyze the methane. Was this chromatograph a portable device, or did you have to take the samples to the laboratory to analyze them later?
Yeah, we transfer the gas to glass bottles and then analyze in the laboratory.
All right, thanks. So the next question is, how many days did you keep the gas in the syringes until you analyze them? So I'm asking this because this has been discussed in our group. So what should be the best way to store the gas in case we have to wait to analyze it? So that's why I'm asking this.
We didn't what are the terms? Sorry, but we transferred the gas in the same moment. We take the gas from the float chambers, and in the same moment we transfer to the bottles.
Yeah.
So and then in the next week, maybe 2 or 3 days ahead, we made the analysis in the chromatography. So in my doctoring, we made this test, how much time we can save the gas in the syringes. So we had a test, but it's not so conclusive, that 1 month we didn't have this variation of the concentration of the gas. But we stopped to use this method and start to use the glass bottles. We think it's more safe to the gas.
Okay, and this is you make like a vacuum in the bottle?
Yes,
before?
Yes.
Okay, thanks.
Thank
and
you.
Congratulations.
Thank you.
Nice, thank you everybody to the questions. Thank you very much for your presentations, Marcelo and Mariana. Really good. Nice to see that more people are interested in the topic in Brazil, and as we have lots of reservoirs here, right? It's really good to see. Let's move to Renske's presentation. I don't know if you want to share your screen, Renske, And as I did not say in the beginning, but we would have something between 2 and 5 minutes for summarizing your main findings. And yeah, and then we move to the questions. Do you want to share your screen or do you want to talk?
Yes, I prepared a very short presentation. But also I want to thank Mariana and Marcelo for their interesting results. It was nice to have something to compare my own results to. And yeah, thanks for that. Let's see. Yeah. So my name is Renske, and last year we conducted a survey of 52 fish ponds in Southeast Brazil. And this was a collaboration between Brazilian universities, a collaborator in the US, and Radboud University in the Netherlands. So this study took place in Southeast Brazil, and in all these fish ponds, we measured greenhouse gas emissions. So we did that using floating chambers to measure the diffusive emissions. And we also installed these bubble traps to assess ablative emissions. And well, this graph summarizes our most important findings. So what you can see here is on the x-axis, the different ponds, and on the y-axis, the methane emissions. And then the yellow bars represent diffusive fluxes and the purple bars represent ablative fluxes. So what's very clear from here is that the ablative pathway was very important for methane emissions in these systems. It was also very variable, so the contribution ranged from 0 to 96%. And this actually coincides quite well, the median contribution, with the study that we did before that Ernandes told about on better assessments of greenhouse gas emissions in these fish ponds. So to summarize our studies, the diffusive fluxes of methane in these fish ponds were rather low. They were higher in the middle of the pond than at the edges of the pond because there was more sediment accumulation in the middles of the ponds. And these diffusive fluxes were probably rather low because the water columns were usually well oxygenated. But the ablative fluxes could be very substantial and have a huge contribution to the total fluxes. So our take-home messages were: take abolition into account when defining emission factors for these ponds, and also low methane emission fish farming is possible. We had some ponds where we had very, very low fluxes. So with the right management strategies, fish farming could be a very sustainable source for protein for our food. Thank you. And I would be glad to take any questions or discussion.
Thank you very much, Renske, for your beautiful work. Really nice to see it again. And I would like to ask the audience if there are questions. Icaro, please.
Yes, nice talk, Renske. Congratulations. It was very clean presentation. I have only one question. According to our PCA analysis, can you say what was the environmental variable that most drove methane emissions?
Yeah, this is still a little bit ongoing data analysis. So it was very hard to tell because these environmental variables were also very variable.
Yeah.
So we had some ponds where we had extreme values of these variables that kind of influenced our models a lot. But we saw in general a trend of high oxygen and high pH having a negative effect on methane emissions. And yeah, also what complicates these things is that these things often go together. So when the pH is high, the oxygen is also high and the algae concentrations are also high.
Yeah.
So yeah, it's still a work in progress.
All right.
And perhaps, yeah, it's hard to unravel these drivers.
Thanks.
Is also interesting because it goes against the general perception that higher eutrophication rates bring more methane emissions, right?
Yeah.
So, but these were all daytime measurements as well. Yeah. That's good to remember.
Exactly, yeah.
Nathan has a question?
Yes. First, I'm very excited about Heinske manuscript. I think we really need this manuscript soon available to the world. And Heinske, because I know the manuscript, my question is not about it. But since you are not from Brazil and you are not from China as well, and these two countries together has like the huge capacity to increase aquaculture and especially to increase the production of fish. And actually, my personal opinion is that we do need to increase the fish production. But we don't know we don't know almost anything about greenhouse gas emission from fish pond yet. We need to generate knowledge about it, especially because we need to know and we need to access all the greenhouse gas emissions from anthropogenic activities. So my question is, what do you think it's more important to advance in this topic? And especially what we have to do to produce knowledge and to increase the knowledge about this topic, especially in Brazil and China?
What I would be interested in personally is to look at different management strategies. So how does it work with drying rewetting cycles, for example? How often should we do that to prevent buildup of sediment, but also to prevent large emissions from these drying rewetting cycles? Of course, the use of aerators would be very interesting to investigate. Yeah, so management strategies probably that are not that technological, so that also small farmers can use those, especially when I look at the systems that I sampled in Brazil. I think that would be interesting to investigate.
Yes, I agree. Yeah, and I'm just thinking a little bit more. I mean, not only the way we produce or the way we manage the systems, but also the kind of fish we produce, right? And also, I mean, the adaptation that we can have to reduce the greenhouse gas emissions. Yeah, I agree. Ernandes, I'm okay with the answer.
We need to have more different techniques, right? To cultivate or to maintain more fish in the fish ponds, right? As Ernandes said, fish ponds, low methane in fish ponds, it's possible, right? It's possible, and as she showed, for example, that we have more emission in the center. This goes to a question also to the previous presenters, Marcelo and Mariana, and because the cages that you found in Ilha Solteira, they were in the middle of the reservoir, right, Marcelo? Yeah, and probably as Renske showed here, you also may have more emissions in the middle of the reservoir, but then. This is something that maybe we could discuss a little bit if it would be possible to place the net cages next to the shore. But if we place the net cage in the shore, we will also put some food for them, right? And then we will also have more carbon being accumulated in the sediment in the shore. Or is it possible, Marcelo, to move the net cage to the edge, or is not possible? Mariana.
Yeah, the net cages were not in the main part of the reservoir. It's rivers that flows to the main river. So it's an environment more not controlled, but with
Stable.
less impact. Yes, less impact than the main river. So this is a great idea to study if we have less concentrations, less density of fish in the same place, could be a strategy to have less impact than the greenhouse gases. So we don't know. We don't know this part yet.
Yeah,
Hey,
nice,
Ernandes,
because one of the most sorry.
And this is a good point, because right now the country wants to increase their fish production in reservoirs. Actually, the government stimulate that from January on. So in the future, we're going to have higher production in reservoirs, and they also stimulate the tanks, the fish ponds. So we definitely need to know what's the best options to reduce greenhouse gas emissions, both from reservoir and from the tanks. And as Marcelo said, most of the production today are in the tributaries, and they are not really in the center of the tributaries, but a little bit close to the shore, because it's easier to maintain, you know, like to work on the cage. So they are not really in the center and a little bit close to the shore.
I'd like to complement the answer, because we have a restriction of deepness. In the producers must have a minimum deep to install the net cages. So we have this limitation because where the reservoir is too, how can I say, hazel? I forgot.
Shallow.
Too shallow. It will be a limit. It will limit the production of the fish production. So sometimes we sample. We had this difficulty to find sites for sampling due to the shallow of the. They have some sites very deep, more than 15 meters sometimes, and this was a restriction to the grab. We use it, but if they install in very shallow areas, there is also a problem.
And there is also a combination of both, right? Because sometimes I see cage inside the ponds, not in the reservoir, but in the tanks, in the fish ponds. Is it like a common practice to have also cage in the ponds?
Here in São Paulo, we mainly near to Campinas, we find more excavated ponds than net cages in fish ponds, in ponds. So that I think that's the case of the last presentation. We worked on a finished project that we had excavated ponds. That is much more common in our region than the net cages.
Renske, did you also measure net cage, just to explain to the audience if you just went to earthen ponds or also measured net cage?
There were a few net cages in my study. I think it's about 3 of them or something. And only one time I measured directly under the net cages. And then I also saw these ablative emissions going into the thousands.
Oh.
So that was very comparable, yeah. It was funny what you just talked about, because I was thinking about a kind of holistic grazing strategy, but then for fish, so that you have the fish in the net cages in the reservoir, but you move them around so that you prevent the buildup of sediment. Would be interesting strategy.
Anyone else?
I have a question to Henske. First of all, congratulations to the group for the study. I think it was very interesting. But I'm thinking about one thing about the bubbles. Do you know if the elevated emissions for bubbles, it's because the high methane production, or the fishes in the ponds?
Sorry, you disconnected for a few seconds, so I missed your question. The high methane production is due to?
So okay, about the
bubbles,
about the bubbles, yeah. do you think the reason for these right bubbles, it's because the right emission, right production of methane, or it's because the movement of the water, because the fishes?
Well, that is an interesting question because actually where I measured these high emissions, where you had the net cages, there were no fishes disturbing the sediment there, but there were very high methane emissions. So I do think there's a large methane production in the sediment in that case, but I wouldn't know for all the cases.
Okay, thank you.
Yeah, you mean because of the bioturbation, right, Marcelo? Yeah. I think
Yeah.
Jean-Pierre, she
Yes,
has a question, please.
Yeah, thank you. Maybe I'm not familiar with the group. May I introduce first or?
Sure, go ahead.
Okay, I'm from Rwanda in Africa. I've just started my PhD research in IHE Delft in the Netherlands. So one of my mentors sent me the link to join in because my project is on the effect of cage farming on Lake Kivu ecology. So I have one question, and maybe it goes with the presentation from Mariana. They presented the results, but I was wondering, because they took samples during they went on the field 3 times, if I remember. So my question is about the changes they observed. Were there differences between those among the periods, like in February, June? I don't remember what was the other month. If they have they observed changes, because I'm thinking maybe there are seasonal factors, but also there is a factor related to the fish feed that they have to provide to the fish based on the age. So my question is whether they observed a change with the time, with the age of the fish and with the season. Thank you.
Marcelo, do you want to answer the question, please?
About sorry, can you repeat the question, please?
If you observe the temporal variability.
Okay, okay, thank you.
Seasonal variability.
This region of Brazil, we didn't see a very higher variations of temperature. The rain is not so intense like the other parts of the Brazil. So our environment along the year was, there was a little variation. So because of this, we didn't see another parameters that can influence this variation of the gases. And probably we saw this strong influence of other variables, like the carbon. So this part of Brazil is not the best part to see the seasonality variation.
Okay, thank you.
Great, thank you for your question, Javier. Is it okay? Yeah, and
maybe with the
sorry.
The other question was if there are variations related to the age of the fish.
Mariana, Marcelo, if you want to say about the age of the fish, there was smaller ones or big ones. Is it, Javier?
We had different phases, but the main phase was, how can I say, after the juveniles, where we had a engorda. How can I say in English? I don't know. it's not my area, aquaculture.
Fattening.
Do you understand? It's
Yeah, the fattening, as Nancy said.
after the juveniles and before the majority of the cases, there was this phase after the juvenile and until the espesca. Sorry, I don't know the term in English.
Yeah, it was before juvenile, and then to the size that they need to be removed from the tanks, right? From the,
yes, yes.
That was the size of them, and then they were there for this period. All of the cages were the same, used the same size of the fish.
Sorry.
All of the cages, they used the same size of the fish, from the juveniles to the big ones.
Yes.
Yeah, and then you did not see any effect of the greenhouse gas from them, right?
Marcelo, for you.
Yeah, we didn't see all the stages of the fish during this period to the remove from the water. So our study used just a few days that we went to the reservoir and measured the emission this day with these variables in the day in this week. So we didn't see this effect of all stages of the fish. So it's like a moment that we measured, see all the parameters, and then we made our analysis.
I think that's a very nice bridge to the talk we're gonna hear later, because that's also what Renske did, right? It's a snapshot sampling. And one of the things we also highlighted in the method review paper is that this is what is often done, but we do not know if this is a good estimate of the total greenhouse gas, because how can you know if you don't have the full picture yet, right? So this is something I think we need to advance in. And that's why it's very nice also that in your newspaper you had the year-round measurements very often in your different ponds, so you can see also this variability along the year.
Okay. Nice. And then let's move to Yumi presentation. Did I say your name right? Please, if you want to present yourself and also give a short presentation between 2 and 5 minutes with some slides, you can also share your screen if you want.
Hello? Can you hear me?
Yeah.
Yeah, okay, I want to start my presentation now. Firstly, I want to introduce myself. I'm from the Institute of Soil Science, Chinese Academy of Sciences. Yeah, I would like to say thanks to Professor Kosten for her invitation and give me such a great opportunity to discuss with all of you. And there's many familiar names in the lecture. And I want to introduce our work in the field of the methane and the natural oxide emissions from the freshwater aquaculture ponds in China. I think Dr. Marcelo, Mariana, and Rinska have give a very good background of this greenhouse gas emissions from these systems. Now I want to start with the aquaculture in China. Yes, as you know, there's more than 50% of the aquaculture production is produced in China, and from 1980s, but this increase is along with a huge converting of the land use. Most of the fish ponds in China is converted from the pit fields. You can see from 1980s, Chinese the area of pit field has decreased very quickly, but the area for aquaculture is increasing. So in 2010, we conducted this research to compare the greenhouse gas emissions from the traditional managed the pit field and the fish ponds. We conducted this work in the basin of the Tai Lake. This is the third biggest freshwater lake in China, and we conduct the greenhouse gas emission measurements in three ponds, and managed measured the greenhouse gas emission with the static chamber method, and we also measured the greenhouse gas emissions in the paddy field. We can see after 12 years.
Sorry to interrupt, but we are not seeing your screen at the moment. I don't know if you, at least I am not seeing it.
Yeah, me too.
You can see?
No.
There you go.
Yeah, now we can see.
Okay, we conduct the research, the study in the basin of the Tai Lake in China. This is the third biggest lake in China, and we conduct established the three marine sites in these 3 ponds, and we measured the GHG emission with the static chamber method, and we also measured the GHG emission in the junction paddy field, and yeah, we also measured the diffusive methane fluxes with this area. We can see after 12 this pond is used to culture this Chinese Nathan crab. I think this is a invasive species in Europe but it's a very famous crab in Southeast China. We can see after 12 years aquaculture, the soil organic carbon has increased about 5%, but we can see that the nitrogen input is generally lower than the paddy field. So now I want to introduce our major findings of our work. We can see the carbon crab ponds has a very static methane emissions especially during the carbon production period, and this methane flux is very high and have a longer period in this period. But the N2 flux is much lower compared with the paddy field, and in the summertime we can see there's some N2 is consumed in these systems, and we find the conversion of the paddy field to the crop ponds. You can see the methane increased about 300%, but the N2 decreased about 95%. So when combine the methane and N2 and the carbon dioxide, we can see that this conversion has increased the net global warming potential by about 245. But for the N2, this emission factor of the inputted nitrogen is much lower. It's lower than 10%. So actually, this system is not a hotspot for the N2 emission. Also, Rinska has reported her findings, but here I want to introduce our findings about the diffusive methane and N2 fluxes. We measured the dissolved methane concentrations in these ponds. We can see this ponds, water in this ponds is it has a very high methane concentrations. It's about 1,500-fold higher than the equilibrium concentration. But for the N2 concentration, it's generally very lower. The saturation condition is lower, and we can also find that this it's lower than the this concentration. But we find the diffusive methane fluxes is much lower than the total methane fluxes measured by the static chamber. But the diffusive N2 fluxes is higher than the static chamber measured fluxes. So we find the diffusive fluxes contributed about lower than 20% of the total fluxes, and the evolution contributed about 80%. But we can see the diffusion flux is estimated by the diffusive model is higher than the actual fluxes. I wanted to discuss. Yeah.
If you want to go for your main, your conclusion, for example, then we can move to the discussion.
Okay. So this I want to show you this slider about the factors influencing the methane and N2 emissions from the carbon ponds. This conversion reduced the redox condition, but increased the DOC, the carbon condition. So, but this condition resulted the accumulation of the ammonia, but decreased the nitrate. So this condition is facilitated to the methane production, but we can restrict the N2 production. So I want to summarize on this stage. Because the feed input increased the organic carbon in these ponds, which facilitates the methane production. The produced methane can emitted through the bubbling, but N2O I think is not produced in the sediments because of the anaerobic condition caused by the carbon input and the anaerobic condition. But the N2 may be produced in the water column. But in the summer, because of the definition in this anaerobic water and in this system, the N2 in the atmosphere can diffuse to this water column and then be consumed by the denitrification in this system. So this is my major conclusion. Okay.
Thank you very much, Yunji, for your presentation, your beautiful work that you have done. And I would like to ask you to stop sharing. Then we, yeah, wow, nice, lots of fish of ponds you have here in a great picture. Yeah.
Yeah, this picture is taken from the southeast China, I think, in Jiangsu province. Okay.
Great. And I would like to ask for questions. Anyone from the audience? Sarian?
Yes, thank you very much for this nice presentation and this extensive work. I have one question, particularly with the question to N2O, which is something we so far have not looked at a lot, but we want to move into that direction as well. And what I was wondering, this emission factor for the crabs is much lower than for the other ponds, right? So the crab pond emission factor for nitrogen oxides is much lower, but you put much more nitrogen in the, well, not much more, but the input of nitrogen in the crab ponds is higher than in the other ponds. So I was wondering, yes, so this one, you see, so the paddy field wheat is considerably lower than the other ones. So I was wondering, what do you think is the underlying mechanism for this lower emission factor for the crabs?
Yeah, yes, as Mariana has presented about the major huge diverse of the methane methane emission from the reservoirs, but we can see the nitrogen impulse is not that's not a big difference from the paddy field, but so I think the big difference of the N2O emission from these two systems is caused by the hydrologic conditions, because in the fish ponds there is about 1 meter to 2 meters water. They produced a very anaerobic condition. This condition maybe can facilitate the methane production, but will restrict the N2 conditions. So we can see the ammonia is accumulated in the crab ponds, but the nitrate is lower than 1 milligram per kilo. So this condition can facilitate the denitrification to complete convert the nitrate to nitrogen and nitrogen gas, but not to the N2O.
Yeah. So it's the depth and the lack of oxygen in the sediment that is probably reducing the N2O emission. Yeah, so that is potentially positive for the other fish ponds, which always have a minimum depth of 1.5 to 2 meters at least. Maybe compared with the plants.
Yeah.
And then I had another, it's not so much, I don't know if it's a question or more a remark. I think for your presentation, it was not explicitly mentioned, but this higher than 100% of the N2O emissions than from the floating chambers. This is based on this equation of John Cole, right? On the gas diffusion coefficient, this K value or not. And I wonder if you did, so that's slide 14, so and I wonder if you did kind of a sensitivity analysis for this, because of course this calculation is very rough, and it's for other types.
I agree. Yes. This calculation is depending on the model using. But we can't measure the N2O emission from the bubbles. So I think I assume this N2O is not produced in the sediments. So I think I assume this N2O is produced in the water column, but not from the sediments.
Yeah, but actually, I mean, this is an estimation, right, on the emission based on and the gas exchange velocity. But this gas exchange velocity is extremely difficult to assess very well. So there is a high range of uncertainty. So this may also be much closer to the 100% than what we are seeing here. Maybe, I don't know.
So I recommend to use the widely used static chamber method to measure this greenhouse gas emissions from the ponds, the lentic systems, so we can have a very stable resource.
Yeah, I think it's of course the chambers also come with problems. No.
Yeah.
But I agree. I think it's a better estimate than on the gas exchange velocity based on this Cole model, which is it has very high or try to have other measures. I know that Raquel and Nathan have estimated the gas exchange velocity in other ways also, and in ponds it may be also doable to add this SF6 or something to try to estimate the gas exchange velocity in that way. Yeah, but nice, very nice. Thank you.
Nice. Anyone else? More questions? I don't know if there are questions in the chat.
I have a question. Yes. First of all, I would like to say very nice presentation. And I was really amazed by seeing the aerial extent of these ponds, because it's so different from what I see in Brazil, from what I've seen in Brazil. And now my hands are actually itching to do a survey in China as well. Would be much less the driving through the jungle. But very nice. And one thing I was wondering is about the management of these ponds, whether there are also aerators that you use, because I saw these white things also in the picture. I was wondering if there are something like aerators. And I was also wondering how the oxygen concentrations were in the water column.
We don't measure the oxygen concentration in this system, but we only measured the redox potential in the sediments about 10 centimeter under the sediment surface. So we can see this is anaerobic condition.
Yeah.
Yes. Yeah.
And
it's
do you know
much lower than the paddy field. This is measured in the
it's very nice to see.
The rice season, so we can see this conversion established a much lower oxygen condition, which can facilitate the methane production. Yes.
Very nice. And do you know if there are any aerators used also in these ponds?
No. Yes, I know another researcher in our city from the Nanjing Agricultural University. They also measured this GHG emission from the ponds. Actually, we conduct this study in under one project. They conduct this study in a very managed crab ponds with the conductor. Yes, equipped with the aerators. Yes.
All right.
So the oxygen is exposed to the oxygen every day, in the morning, the afternoon, every day. Yeah. So they find that the methane emission is much lower, and until it's increased. So we can see the diverse methane emissions from Marcellus work. We can see very diverse. So I think this diverse is driven by the oxygen condition, so the flow of the water or something. Yeah.
Thanks a lot.
What else? More questions? I cannot see everybody. If anyone is raising the hand.
If there's no more question, I would like to ask Meredith, how did you test this better gas exchange with the different models? Because what we are planning to do is we want to sample a lot of ponds in Brazil in different biomes, and of course we want to use the chamber methods, but to get more data, maybe we will also get snapshots of only concentrations, and then it would be nice to have a kind of a best fit possible to extrapolate this to fluxes as well.
Yeah, so we are looking at this for a metabolism project where we have some sites with wind data, some sites without. So we're not thinking about it from the greenhouse gases right now. But with that, we're comparing sites that have kind of all of the measured data and sites that aren't. And we're looking at different lake size or pond size classes. They're all small water bodies. And I think this is it's a student who's on their committee. I think they found coal was overestimating what that gas exchange value would be. And we compared it to, I had a student during my PhD who I was advising, and she added propane to a series of small ponds, and we actually measured the loss of propane over time to get the gas exchange rate. And the Vashon and Prairie model includes the wind speed and the surface area. And at low wind speeds in small water bodies, that intercept is pretty close to the value that we had found in the ponds. And so I think what he did to compare was use the constant value that I found for like the average of those size classes, compared that to the Vashon model, and compared that to the coal model. And I think the coal model was like much higher than those other two. And so now we're at the stage where we're trying to decide if we need wind speed, and some sites have wind speed and some sites don't. And what would happen if we just assume this constant K versus use the Vachon and Prairie model that includes measurements of wind speed. And I think what we're finding, I'm trying to remember our last conversation, I think it depended on if sites were more exposed or more sheltered. And if they were more sheltered, then the constant value was just as good as the other model. But if they were a little bit windier, then we might need those wind speed measurements. So for sites like this, like I'm not familiar with what all the fish ponds look like, but on slide 17, that's just this big, vast, open, exposed landscape where I imagine there's a lot of wind. I might be wrong, but in that case, you might need to have that wind measurement to get a better K estimate.
Mm-hmm. I guess you have K because for John Cole's model, you also needed that, right?
Yeah.
Or did you use one fixed wind value, Yunji?
Yeah.
No, I was wondering if you took actual wind data or if you took an average wind speed, for instance.
Yes, we measured the wind speed. We have a station in jump to this pond, so we calculated the every point of the flux with the wind speed in this part. Yeah.
Yeah, so I imagine for the Brazilian fish ponds, we could of course also measure wind speed when we are there, and then
yes, this study is conducting a experimental station of our Chinese Academy of Sciences. So the equipment of this station much better, very good. So we can measure this.
Yeah.
Parameters. Yeah.
Nice.
Great. Any more questions, or shall we move to the end? Yep. Yeah, I thank everyone to be here. Thank you very much to be here. It was a really nice session talking about fish ponds and greenhouse gases and many other things. Thank you very much for everybody to be here. I would like to pass to Sarian and Nathan if you want to say something. Thanks.
I just want to say that we have a lot to do together in the future. We have good options to collaborate, and it's good to have this session here where we can know each other and we can plan like some cooperations in the future. So thanks for being here. It was a big pleasure to participate.
I do not have a lot to add. I think we have a lot of plans, and it's nice to have this contacts also to see if what we find is something extrapolatable or if it's something very peculiar. That is, of course, very nice to know. I would like to make a picture also of us as a group. Who knows what this is bringing us at some time? So I don't know if Priscilla and Jonas would also like to turn on their camera.
Sure, I'm not part of the group, but I can be in the picture.
Yeah, of course you are part of the group. No? I know that they have a lot of nice ponds in the surroundings of in Natal as well. So you can.
Well, I would be the one criticizing all of these ponds and aquaculture and tilapia and everything. So I'll just be quiet here.
No, but you can make them better, no? Okay, here we go. 1, 2, 3. Thanks.
Nice. Thank you very much. And then here we leave in the afternoon. We have a workshop.
Yes, let me just say it again for the ones that are still here, the young ones. This afternoon we have our last meeting. Sarian will be there. It's just an hour for the early career. It's a moment there where we can meet some of the keynote speakers and some of the big names you all know from the literature. So join us at 1 o'clock in 1 hour, less than an hour. Thank you.
Thank you. Thank you, Ernandes, for heading the session.